Good morning, good afternoon, good evening, and welcome to this next webinar in our chromatography series. I'm very pleased to present, I'm not presenting, Tony's presenting. I'm very pleased to introduce Tony Edge, who's going to be presenting today's webinar on SPE protocols. Firstly, just a bit of housekeeping. You are able to ask questions during the presentation. There is a questions box that you can find in the GoToWebinar menu. So please type your questions at any point during the webinar, and we will either respond during the session or if we've got time at the end, we can answer some questions as well. If we don't have time to answer all the questions during the session, we will come back to you by email. So please feel free to keep asking questions. So as I mentioned, I'm really happy to be joined by Tony Edge. Tony is our R&D manager at Avantor. He's heading up a team of specialist scientists in manufacturing and developing next generation stationary phases for HPLC. He's worked in both manufacturing and also industry, having periods of employment at LGC and also AstraZeneca, as well as Thermo Fisher Scientific and latterly Agilent Technologies. In 2008, he was fortunate enough to be awarded the Destiny Memorial Lecture for his contributions to innovating separation science. And in the same year, he also won a Clinical Excellence Award from AstraZeneca. He's currently the president of the UK Chromatographic Society and also the chair of the Permanent Scientific Committee for the ISC, as well as being the chair for the next ISC, which is due to be held in Liverpool in 2024. With that, I will pass you over to Tony, and thank you very much.
Hi, and welcome to today's webinar, which will be looking at developing an SPE protocol for the analysis of biological samples. Before I begin the webinar, I'd just like to thank my co-workers, in particular Matt, Colin and David, who have helped in producing the bulk of this data. So let's begin this journey then. We'll initially start off by looking at a quick introduction to sample preparation and why this technique is so important and its applicability across a range of different biological sample types. Then we'll move on to specifically looking at some of the challenges associated with oligonucleotides. This is a therapeutic area that has seen a lot of interest, in particular since the rise and fall, fortunately, of COVID. We'll look at why these compounds are so difficult to analyze and some of the problems that lab analysts have in developing their methods and also maintaining their methods. From there, we'll then move on to the chromatography and how we develop chromatography for oligos. Then move on to solid phase extraction optimization, looking at how to optimize a method and then making that approach applicable to the development of a method for oligonucleotides. In this journey, we won't reach the final destination. What we will find is an approach that will be very applicable. So we'll be looking particularly at the chemistry involved in extracting oligonucleotides. And from there, we'll then wrap on conclusions. Sample preparation is a process by which we can convert complex samples, such as blood, plasma, food, etc., to a form that would be suitable for analysis on a chromatographic system in this scenario. It has significant advantages. It reduces the matrix components and also matrix interferences. This allows us to get better sensitivity, improves the instrumentation and column lifetime, and because it's a less complex sample, means that we can now do faster analysis. We've got less things to separate. This really is being driven by a variety of different industries to try and reduce the analysis time and therefore to think about how we can apply sample preparation in a manner that is going to be beneficial to these different industries. So the demand is coming from bioanalytical laboratories, clinical forensics, toxicology, drug discovery labs, for example, where there is an increasing number of samples due to the way that we live our lives. We all want to be healthier, better, and as a consequence, we need to do more analysis. And this, in term requires a fast, simple sample prep strategy. When we look at the traditional workflow involved in LC mass spectrometry, we can see that we have solvents that move into some form of HPLC pump that then flow through a high performance liquid chromatographic column and then ultimately into the mass spectrometer or detector and then finally onto some type of data acquisition system. In order to apply samples that will be compatible with this format, it is necessary in a lot of occasions to do some form of sample preparation, and the box here shows how sample preparation fits into that nice little LCMS workflow. If we're looking at the analysis of a bioanalytical sample, then there are a variety of different common approaches that can be employed. Probably the most common is protein precipitation. This is used as a fast, quick technique and involves the addition of some form of chaotropic reagent to your biological fluid. So typically a 3:1 ratio of acetonitrile to plasma with the addition of some form of internal standard. This results in the formation of a precipitate. That precipitate can then be either filtered or centrifuged so the remaining supernatant can then be injected onto your LCMS system. Syringe filters can also be used, and this removes particulates from the sample. One thing to be aware of is that it doesn't remove proteins, and so therefore this technique might need to be used in conjunction with other forms of sample preparation when dealing with bioanalytical samples. Supported liquid extraction is an alternative technique to liquid extraction and utilizes the immense surface area that you have with diatomaceous earth, a biologically derived form of silica, to ensure that you get good mass transfer between an inherently hydrophobic phase and an inherently hydrophilic phase, if you like, some form of organic solvent such as hexane and some form of aqueous solvent such as water. The way that this would work is that you would mix some of your sample, some of your plasma sample with water, add some internal standard, apply it to the cartridge of diatomaceous earth, and then add 1 ml of some form of water immiscible solvent. In this case, we've exemplified ethyl acetate. The final form that I'm going to mention as a common approach is the use of solid phase extraction. And there are a variety of different solid phase extraction cartridges available. I've just highlighted some of the silica-based ones from JT Baker Bond. In the next section, we will go through in some detail some of the approaches that can be used to optimize a solid phase extraction method. But why should we do sample preparation? Why not just inject that fluid that is plasma or blood onto a HPLC MS system? Surely it'll be okay. Well, unfortunately not. No. What we find is that the sensitivity of the detector will drop off very considerably. Some of this is because we made the assumption that the detection system will be selective. And with mass spectrometry, that's simply not the case. We find that ion suppression can occur within the source. This reduces the signal output, therefore masking part of the signal from the analyte that we're interested in dealing with. As well as this, we can get other matrix interferences, and indeed we can get protein binding if there's proteinaceous matter within the liquid sample. System robustness is another thing. Injecting plasma or blood onto a standard HPLC system will degenerate the columns much quicker. Typically, you might get 20 or 30 injections if you were to inject neat blood onto a HPLC column. It will also mean that there's less maintenance required on the detector as well, which obviously benefits to that. In terms of in the injector, the syringes are less likely to block. There's also less likely to be contamination, and this ultimately will give you a more robust system, a more reproducible system. Sample preparation can also allow you to concentrate the sample up. So it means that if we're dealing with large volumes of substance, large volumes of blood or plasma, or potentially urine, it means that I can start to look at very low levels of either endogenous or exogenous material within the biological system. It also reduces the sample complexity. So in terms of my chromatography, peaks to separate. we may not always want this, particularly if we're doing some form of omic analysis, where we're really interested in understanding the full profile of those individual biomarkers that are present within that biological system. And this chromatogram really does highlight the challenge of complex sample analysis. So I'm very grateful to my colleague, Professor Ian Wilson, who supplied the attached chromatogram that shows what happens if you inject neat mouse urine into an LC-MS system. The number of peaks is immense, and what we see is that these very, very complex samples can have literally millions of different components, which chromatographically would be almost impossible to resolve. The use of mass spectrometry will allow us to do focus analysis, but we may not be able to see the matrix components. And co-eluting peaks can and do interfere with the ionization process, whether they be charged or indeed neutral. And so if we look at trying to use complex samples when analyzing with LC-MS, we will have complications. What we also tend to find is that the chromatography in a bioanalytical environment tends to be focused on an individual analyte. And so we don't actually monitor the chromatography of non-targeted compounds. And again, this can be very detrimental to the overall analysis. When dealing with complex biological samples, there are many factors to consider, and I've listed some here, including protein binding, ion suppression, adsorption, solubility, recovery, carryover, and reduced column lifetime. All of these challenges have to be addressed when we're developing a method. For simple small molecules, these challenges are very tough. As we start to increase the complexity of the analyte, we begin to enhance these challenges. And so when we're looking at oligonucleotides, for example, these challenges become very extensive and very difficult to overcome. Another challenge that can often happen, particularly when we're looking at small bed weights, is associated with the process of manufacturing. And so when cartridges or deep well plates are filled with the substrate, oftentimes that substrate may not be packed effectively, resulting in channeling, or indeed if the frit isn't put in properly, we may result in voiding. And then with the smaller bed masses, it's not always possible to accurately weigh out that bed mass every time. And so we can get differences in the bed weight. One approach that can be utilized to overcoming this is to, instead of using loose packed material, is to use a composite silica, which can then be applied into the SPE tube or deep well 96 well plate. And because we're now using something that is very well defined in terms of the manufacturing and indeed the size and the volume, it means that we don't have any channeling. There is no voiding, there is no bed compression, and there is also a consistent bed weight. The impact of using a composite SPE can be clearly seen on the current slide. Here we're using 3 comparable stationary phases, but in 2 different formats. The composite SCX versus competitor A and competitor B. If we initially look at the RSDs in the middle box, we can see that the RSDs are fairly comparable and all are very low. And so initially we'd see perhaps there's no significant advantage of using the composite SCX versus the competitor A or competitor B. But where the issue then lies is that when we start to look at the detail, and so if we look at the plate itself, we can start to see with competitor B in particular that actually not all the samples are passing the current ICH M10 guidelines. And this means that if you're running real samples, there is a potential that you might not be able to get the right results. It could even potentially result in a failed batch, and that is a very costly experiment to have performed. And so using the composite, we can clearly see that the recoveries are all green and there are no issues. There's no well-to-well variation. competitor A and competitor B, we occasionally start to see individual wells that are not performing at the level that they should be. And this is using a relatively simple methodology. Methodologies that require more additions of washes, etc., potentially would have significant differences, or even on the data being performed here. Clearly then, there are significant advantages for using the composite material over a loose packed material. But what then about the method development considerations? At the start of the presentation, we looked at a variety of different sample preparation techniques. Each of these techniques has advantages and disadvantages, and some of them will be more applicable in different types of analytical environments. So the number of samples, detection limits, the complexity of the matrix compound will all have decision-making implications in terms of which of those different techniques to use. As well as that, the analysis time available, the physicochemical properties, and also financial implications. In terms of the method development approach, one of the first things that should happen is to have a look at the structural and physicochemical properties of the molecule. So in this example, we've taken propranolol, a beta blocker developed by AstraZeneca/ICI. This is a basic compound, and we know it's basic compound because as we change the pH, we can add or remove a proton to it to generate a charged or an uncharged species. So at low pH, the molecule is charged, and at high pH, the molecule is neutral. The point at which that changeover between neutral and charged state exists occurs around the pH, which in this case is around 9.5. This also tells us, gives us an indication of when we can run the chromatography. So ideally, we don't want to be running near the pKa. There are too many changes in terms of the charge state of the molecule, which is in a dynamic equilibrium. It also gives us a good indication of the design of the mass spectrometer detection parameters as well. So where we are adding a proton, that suggests that this molecule will ionize in a positive charge environment. If we were to do some modeling on this, then we could determine the log D or the log distribution. This gives us an indication of the type of chromatography we should use. Negative or very low log Ds, we might prefer a Hillick type approach. For log Ds that are above 0, we might prefer a reverse phase approach. In terms of the extraction, this also gives us good indication of the approach to use on the extraction. Charge states are very good with charged SPE media or mixed mode media. It can also give us an indication of the type of mechanism, hydrophobic or ion exchange. As well as this, it can also highlight potential issues. So the pH to avoid I've already discussed in terms of the chromatography. But if we've got high log D, that may give an indication that this compound has a lot of carryover. But equally, a lot of charge states may give an indication that this molecule may stick to a lot of different charge components within our analytical system. For basic compounds, if we can see that we're analyzing on a silica column, this may give an indication of peak tailing, so we can start to choose higher purity silicas. It may also give an indication of the recovery of the molecule from any form of extraction process. And all of this is critical in developing the final methodology. So can we use this approach with the analysis of oligonucleotides? So initially, what are oligonucleotides? Oligonucleotides are the polymeric form of a nucleotide. So what's a nucleotide? A nucleotide is an organic molecule that comprises of a nucleoside and a phosphate group. A nucleoside is a nitrogenous base bound to a pentose sugar, in this case a ribose or deoxyribose. And we can see here the available structures that are available for making up those nucleosides. And it should be noted that there are a finite number of structures that form the basic building blocks of a nucleoside and hence an oligonucleotide. In terms of compounds and the naming of those compounds that form a nucleotide, the nucleoside is the basic building block. And there are two forms. There are purine and pyrimidines. If the compound contains a purine, then the ending is replaced with ocene. If it contains a pyrimidine, then the ending is replaced with idine. And this then leads to the structure of a nucleotide. So in this example here, we have a nitrogenous base, which can either be a pyrimidine or a purine group, as described previously, attached to a 5-member sugar group. So that will either be a ribose or deoxyribose. And then finally, to make up the nucleotide, we attach one or more phosphate groups. One of the parameters that I discussed briefly beforehand was the importance of log D, or log distribution. The log distribution refers to how a molecule distributes itself between two immiscible phases, typically octanol, so in this case that's the little O, and water. These two phases are immiscible. And what we can do is we can have a look at individual properties of the molecule, which we can derive through basic principles in order to gauge what type of interactions those individual components of the molecule will have, but also then to gauge what the overall log distribution factor will be. And this can help us in designing our chromatography, but also potentially identifying any issues going forward. And we can apply this approach to the analysis of oligonucleotides. It should be noted that log D is a composite polarity descriptor, and it combines a variety of different types of interactions, ranging from interactions associated with the N and pi electrons, dipole-dipole interactions, hydrogen bond interactions, van der Waals, London forces dispersive interactions, and then for charged molecules such as oligonucleotides, also looking at ionic interactions. So if we then do this approach on a very simple nucleotide, so not even an oligonucleotide, what type of information do we get out? So here we've got a very simple molecule. There are packages available that can analyze this and perform log D calculations. So in this case, we're using a package called Chemicalize. The web address is given there. And what this enables us to do is identify ionizable groups that will change as we vary the pH. Becomes very evident if we compare the plot of the distribution of those different groups with the propranolol that we saw earlier, that this is much, much more complicated. And remember, this isn't even an oligo. This is just a simple nucleotide. This is one of the building blocks. We can see that as we vary the pH, we see a range of different forms of the oligonucleotide appearing. And across the pH range, there may be regions where we only see one existing, but they're very, very tiny areas in terms of that pH range. We can then start to look at the log distribution factor. And again, we start to see some interesting facets coming from this. The first is that the log D is very low. It's negative, -4. This is a logarithmic scale, remember. So this now means that it's highly charged, and the chances of performing reverse phase on this type of molecule are minimal. So we would have to look at a different type of approach. We can use reverse phase, but we'd need to use a different form of reverse phase in the form of ion pairing. What we also see is that as we increase the pH, the log D becomes more and more negative. And the reason for this is because as we change the pH, we're activating more of those charged states. In particular, increasing the pH, we are removing protons from the chemical system, and that means that we'll end up with more acidic groups becoming charged, which is exactly what we see. This is problematic because all of those charged groups will interact potentially with charged surfaces, and on a chromatographic system we have to be very aware of that. If we're looking to run our chromatography on silica, silica is a charged species. It will potentially interact in some form with these type of molecules if we're not very careful. We also need to be aware that there are very few stable regions for that log distribution. So small differences in the chemical environment that may occur because of temperature, subtle changes because of local pH variations, because we've got a larger concentration of a particular analyte or matrix components, can also have an impact. So all of these things we need to be aware of when we're developing the assay. This makes it a lot more complicated than, say, for the example of the beta blocker that I showed earlier. On the previous slide, we looked at the log D, and we also saw a plot of those different components. This slide shows those components, but now we can actually see what the structures are. And we can see that each of these structures in themselves, from an electrostatic perspective, are quite complicated. We have multiple charge states. We have Zwitter ions, amphoteric ions that will cause us complications when we're looking at developing an assay. This means that the analysis of oligonucleotides is very complex and something that we need to be aware of when we're developing the methodology. And so the approach that we'll be talking about towards the end will give us an introduction in the parameters to look at and to investigate to ensure that we've got good, robust, stable bioanalytical workflow. So let's go back to that log D plot and have a closer investigation. I mentioned previously that the log D was -4. -4 is very, very polar. This means the chances of retention on a traditional C18 phase, a very hydrophobic phase, would be minimal. We'd have to be looking at some other form of chromatography. The compound is always going to be charged across that full pH range. Also, as we noted previously, the log distribution factor is very variable, and we have multiple interaction modes, different types of interactions, multiple charge states, regions where we've got a degree of pi bonding, regions where we've got a degree of van der Waals types interactions. This means it's very, very complicated in terms of developing a robust methodology. The approach then that we use for the chromatography is typically through ion pairing chromatography. This is a technique for retaining, separating ionizable solutes on a reverse phase material such as C18. Typically, it's preferred over ion exchange chromatography, which uses the principles of interactions between oppositely charged electronic states, because we get higher efficiency and we can also hopefully get greater control over the selectivity. In order to work, the mobile phase will contain a hydrophobic counterion, often referred to as an ion pairing reagent. So it provides retention for very hydrophilic ions, gives better control over retention factor for mixtures of acids, bases, and neutrals, and is a powerful means of controlling selectivity. However, the mechanism is still a little unclear. The compatibility with LC mass spec, which is the traditional approach for the analysis of biologically derived samples, is not so good. It's not compatible. However, ion pairing reagents such as TFA, HFBA may provide a route forward there. Typically, it's slow equilibration of the columns, and classically there are substantial memory effects with this approach as well. So how does it work? As I say, the explanation isn't 100%, but this will give an idea of how it works. So for ion pairing chromatography, we typically use a bonded stationary phase, so a C8, C18, for example. So if we look at the top left figure, this is what would normally happen with a protonated molecule. The same would apply to a deprotonated molecule like an oligonucleotide. So we see very little interaction because there's no level of ion exchange occurring. So what we can do is that we can add a charged sulfonic acid to the mobile phase or other ion pairing reagent. And what then happens is that this charged molecule, which has a hydrophobic component and also a charged state, is retained dynamically as a coating on the hydrophobic stationary phase, and we now start to see interaction with the charged molecules. We can also see the converse of this is where the ionizable molecule itself interacts with the oxygen. So quite a complex series of interactions occurring that are very, very dynamic. So equilibration becomes very, very important with this type of technique. Looking at the structure of an oligonucleotide, and here we've just zoomed in on a small proportion of a thymidine oligonucleotide. We can see that the abundance of phosphate groups means that this becomes an ideal technique for ion pairing chromatography. There would be little interaction with the standard C18 reverse phase, but the interactions, the polar interactions that we get between the phosphate and those ion pairing reagents will ensure that we get good retention and therefore good separation as well. And here's an example of how we can run on a mass spec looking at the analysis of a range of oligonucleotides. So in this case, we're looking at a 15-mer through to a 40-mer. The column we're using is an ASXL 1.7 micron, specifically designed for oligos. It's 100 by 2.1, and note the complexity of the mobile phases. The gradient method itself comprises of a 25-minute run with the bulk of the components from the test mixture eluting prior to 10 minutes. We see that we get very efficient chromatography, the peaks are very sharp, and as we increase the mass of the molecule that we're starting to analyze, we start to see some finer structure appearing of some of the impurities that get generated when we're producing the different oligonucleotides in this particular series. So we've now got our chromatography. Now we need to start focusing on our extraction methodology. And so in this case, we'll be using solid phase extraction. So the method development is relatively simple. I'll talk through a process where we can go through and optimize the chemistry relatively quickly. There are quite a few manual steps to perform for the analysis of oligonucleotides. So in this presentation, we'll give you an introduction to some of the developments of the chemistry, but there is still more to be done in the method development. If you like, there's a part 2 coming up. It is more expensive than protein crash, but in this case, we need that selectivity, and so we'll be applying the brute force of solid phase extraction in order to get that. Matrix removal is good with solid phase extraction, and it can be applicable to a wide range of compounds. So particularly oligonucleotides, we can talk about the phase selection for that. We know that the phosphate group is charged, has a negative charge on, so some form of interaction with that positive charge would make sense. So a positively charged stationary phase, something like an anion exchange, would make a lot of sense. And most of the work that we're presenting today will be looking at the 96-well format. So how do we go about optimizing the performance of an SPE methodology? In order to do that, we really need to understand the different steps involved. So the traditional steps associated with solid phase extraction, and there are 5 of them. There's conditioning, and at this point what we're doing is that we're wetting the cartridge. The bulk of the surface area is within the pores. If we don't wet the cartridge appropriately, and what we mean by wetting is getting liquid into the pores, then it means that we lose the bulk of the surface area, which means that we'll overload the cartridges, the phase, very easily. We can do this in a variety of ways. Typically, when we're looking at some form of bonded silica, we may traditionally want to apply some organic solvent. The organic solvent will not have any negative interactions with the stationary phase and therefore will fill into the pores. An aqueous solvent, when we're looking at a bonded phase, may well get repelled from the pores, and so therefore we'd lose active surface area. So the conditioning step is important for activating all of the surface area ready to get our sample attached. However, typically the solvent that we use on that step will be quite a strong solvent. What I mean by strong is it will easily elute our compound of interest off. And so what we then do is we equilibrate the phase by removing the bulk of the organic solvent that we've already applied. We still got liquid in the pores. There's still a small amount of organic solvent that allows that to happen, but the bulk of the solvent will be a very low eluting power solvent. What we then do is the most critical step, which is loading the sample onto the cartridge. In this case, the type and nature that the sample is in becomes important. So we need to make sure that we've modified the pH of the sample so it best interacts with the stationary phase, or vice versa. If we're using a stationary phase which can be switched between different ionization states, it's important that we get the right ionization state. We apply the sample at a relatively low flow rate. The idea here is that we want to give the sample the opportunity to interact in a favorable manner with the stationary phase. So when we talk about sample, we're actually talking about the analyte. We want the analyte to interact. And so if we're looking at charge states, that means giving the molecules the right orientation and the time for some type of interaction to occur. At this point, we start to lose some of our matrix components, which is a good thing. What we then do is we look to then apply a wash step. This is where we try to remove more of the matrix as possible. The typical approach will be to look at a slightly stronger eluting strength wash solvent that elutes our matrix components, or some of our matrix components, but doesn't elute the compound. And I can talk through how to optimize that later on. And then the final step is to then apply a solvent which will elute our compound, our target analytes, but ideally only elutes those compounds. So we still leave other remaining matrix components on the cartridge. And by applying this process, we should have a fully optimized method. Now, clearly, in order to optimize this, it will require some effort. One of the first things to consider is the selection of the sorbent. So for oligonucleotides, I've already mentioned that they have a variety of different interactions that they can have. So if we look at the different types of interactions that SPE sorbents have, they can have ionic, polar, or non-polar. Stronger retention is provided by ion exchange. That may potentially give better selectivity because it allows for the removal of more matrix components potentially. If we can combine interactions, that should give us better selectivity. In terms of the range of different SPE phases, there are a broad range, and this flow diagram gives an overview of how to go through to identify what type of phase should be used for what type of compound. So if our analyte, for example, is water soluble in ionic, then we'd need to determine whether or not it was an anion or cation, and based on that, we might then look at using a strong anion exchange, a weak anion exchange, or a strong cation, a weak cation exchange, for example. And we can go through this in order to identify which is appropriate. For oligonucleotides, we're looking at an ionic compound that has anion characteristics, and so predominantly be looking at either SAX or WAX or a mixed mode of those. So in this case, we'll be actually looking at a weak anion exchange. So when we're looking at those 5 different steps, there are several considerations. The conditioning step, as I mentioned previously, needs really to be a strongly eluting solvent. The rationale for that is that it disrupts the type of bonds that are occurring, but it also gives you the best interaction with the surface of the stationary phase. So that means we can wet the stationary phase. We also need to apply a suitable volume. Typically, we'd apply the volume of the cartridge. So if it's a 1 ml cartridge, we might apply a ml, for example. Most of the times, the volume that we'll be using will be massively in excess of the bed weight. The equilibration stage, we need to make sure that it's a weakly eluting solvent, and again, a suitable volume that will displace that strongly eluting solvent. In terms of the load stage, the ionization state becomes important, so we need to make sure that we've modified the pH accordingly. We also need to be aware of the bed weight and make sure that we can choose a suitable bed weight for the type of sample that we'll be applying and for the concentration of the analyte that we're looking for. We also need to be aware of the cleanliness of the matrix as well. because that will potentially take up some valuable surface sites on the surface of the stationary phase for the solid phase extraction sorbent. When looking at the washes, the allotropic strength and the suitable volume also become important, so multiple volumes. In terms of the elution, which is one of the things that we'll be focusing on later in the presentation, the allotropic strength of the solvent is important, and also the volume, but also the nature in which we apply that volume. Do we do 1 ml in one go or 2 lots of 500 microliters? As we'll see, that becomes important in the following slides. At the start of this process, I said that developing an SPE method is relatively quick and easy. Okay, I need to justify that. So what's the process? The process in this scenario is to take a single cartridge, condition and equilibrate in accordance with the manufacturer's instructions. Using that same single cartridge, we would then load on our analyte of interest. In this scenario, we're not loading on matrix components, so we're just looking at the chemistry. I would then capture into a vial or a test tube the contents of that load. And then what I then do is gradually apply to the same cartridge, increasing allotropic, greater eluting strength solvents, and then capturing each of those eluents as they pass through the cartridge. And by doing that, I can then determine where the compound of interest comes out. So in this case, we can see that at low elution strength, we get no analyte occurring, but as we increase the elution strength, at some point, the analyte will be removed from the cartridge. And if it's not removed from the cartridge, then clearly it's inappropriate. In terms of the analysis, we're only looking at qualitative data for a single compound. So in this scenario, we can use flow injection analysis. We don't need chromatography. We're typically only looking at one compound. So this means it's a relatively quick and simple analytical process. So what does the data look like from these elution profiles? So the top left gives the raw data. And what we can see here is a plot of the response from the mass spectrometer versus the different compositions that we're applying through the SPE cartridge. So on the load step, we don't see anything coming through. That's good. We don't want to see breakthrough occurring. And then as we gradually increase the methanol concentration from 0 in the first wash, which is 100% aqueous, to 20%, 40%, we can start to see some components coming off. At 40%, we start to see the first amount of component coming through. It's very small. So at 40%, that starts to be where we start to see things coming off, and then very quickly it ramps up. But it's also very evident that as we increase to 100%, it's taken several washes at 100% to fully get rid of all the analyte from the cartridge. So in terms of choosing what the wash solvent would be, in this case we could probably get away with 40% methanol wash. In terms of the elution in this case has to be 100%. But what this chart is telling me is that actually we may need to do multiple elutions or use a stronger solvent in order to elute that compound off the cartridge. So let's now look at thymidine 20mer, and this will be the molecule that we're starting to develop the method on. So here we've got the chromatography. We're using an ACE 3 micron super C18, 50 by 2.1. We're only looking at one compound in this case, and we get nice chromatography occurring within 4 or 5 minutes, with a total run time of 12.5 minutes, allowing us to analyze more components if we want to. We're running in negative mode on electrospray. We're looking at several different transitions so that way we can capture fully the oligonucleotide as it comes through. In terms of the SPE method, we'll be using a weak anion exchange. In this case, it's a loose filled material. The conditions for the sample are given. The conditioning and equilibration as to manufacturer's instructions, and we'll be focusing on the elute state. So at the moment I've specified 50 millimolar TEA in a variety of different solvents. And what we want to do is really look to try and come up with some hypotheses about what will happen, and based on that determine what the experimental parameters should be for the final methodology. And as I say, we'll be focusing on just the elution step initially. So in this case, we've hypothesized that we want to optimize the methanol water. And as we can see, when we're looking at varying the methanol water, if we apply our control, which is equivalent of what should be eluted from the cartridge, we can see as we varied the methanol water that actually we're clearly not eluting everything off. The idea was that increasing the methanol would break some of the hydrophobic interactions between the oligo and the wax material, but this clearly didn't happen. And so actually increasing the amount of organic in this case is detrimental. So we then started to look at saying, okay, what happens if we decrease this? So let's decrease the amount of organic and see the impact that that has. So still using the 50 millimolar TEA, and now we can see that actually we're getting much higher return with the 100% aqueous. So we're not getting any breakthrough, which is great, but we're getting 60% recovery using the 100% aqueous. So the next stage is then to start investigating the impact that TEA has. So the original method used 50 millimolar. So now we're looking at using a variety of increasing concentrations. So 100 millimolar, 200 millimolar, 400 millimolar. And as we increase the concentration, we start to see that we get increasingly higher and higher recoveries, with the best recovery coming from 400 millimolar. Previously, I'd mentioned that it wasn't necessarily just the volume, but also how we eluted it. So if we did it in one elution or two elutions. And to this effect, the following data identifies that. So here we have a graph that looks at a control sample, a solution of compound in 200 millimolar TEA, and all the responses should be 100% of that from the elution. We can see that in terms of the first 500 microliter solution, we have about 60% of the 20-mer thymidine oligonucleotide coming off, with 40% coming off roughly in the second elution of 500 microliters, and then the third part we get a little bit remaining. If we have 4 lots of 500 microliters of 200 millimolar, we can see that we get pretty much 100%. Moving to 600, 6 times 500 microliters, we still end up of 100%. So this would suggest very strongly that multiple elutions would be better in this case. And just to further emphasize this point, here we can see the difference between eluting 4 times 250 microliters versus 1 by 1 mil. And we can clearly see that doing 4 lots of elution gives us better recovery overall on that final elution step for that oligonucleotide. The next stage was then to investigate the difference between the different concentrations of the eluting solvent in TEA. So in this example, we're looking at eluting with either 200 millimolar of TEA or 400 millimolar of TEA. And to emphasize the point of about multiple elutions, within each of those sets, we're looking at a subset that looks at differing number of elutions, albeit the same total volume. So we're either looking at 4 times 250 or 2 times 500 microliter elutions. And what we see is that in all cases, splitting the elutions increases the number of the amount of recovery. 400 millimolar TEA in the final aqueous solution solvent does increase the recovery though from 79% to 100% and slightly more indeed. So at this point we thought it appropriate just to check accuracy and precision using aqueous solutions going through the extraction process. As an internal standard, we've chosen an acid, so in this case it was salicylic acid, and we've run over a range from 300 to 30,000 nanograms per mil, which we found was pertinent for this particular compound. Duplicate injections were done, and the results in the table represent the average accuracy from duplicate injections. We can see from the data points that the precision is very good. Indeed, all of the data based on n equals 2, the precision is less than 3%, which is excellent. And in terms of the accuracy, again, the accuracy is very, very good, which suggests that in terms of the methodology that we have, in terms of optimizing the chemistry, we've actually progressed quite a substantial way. There is still more work to be done, but certainly we've demonstrated that the conditioning, the re-equilibration, and the loading stages in terms of the chemistry, and then the final elution stage give us highly accurate and precise data for this thymidine 20-mer. The approach that we've taken is such that it could be applied to other molecules, with the idea that we've highlighted critical parameters such as the amount, the concentration of the final elution solvent with regard to the ion pairing reagent, in this case TEA, also the number of aliquots of elution solvent that is applied, and ensuring that we have the right mobile phase. So in this case, we've identified that water is better than any water methanol mixtures in terms of optimizing the elution. So overall, we've started to really focus on the pertinent chemical points, the chemistry that is going on between our oligonucleotide and our stationary phase molecule that comprises within the SPE media. And this approach could be applied universally across the range in order to optimize the chemistry. At this point, we wanted to start investigating the wash solvent. In order to characterize where we were, it was felt pertinent to take a plasma blank through the current extraction process, which contains no wash step. So we would anticipate that we might remove some of the components, but certainly we wouldn't remove all of the matrix components from plasma because we've simply not optimized that wash step. So what we did is we monitored for a particular matrix component. So in this case, we were looking at a series of phospholipids. We're looking specifically at 5 phospholipids, and the masses of those 5 different phospholipids are given in the text. So 496, 524, 704, 758, 806. There was no need to tune up on those masses. The phospholipids are present at relatively high concentrations, and they are very, very ionizable. So it means that using mass spectrometer, we can pick them up very quickly. What we've done here is that we've taken 6 blank plasmas. We've taken it through the extraction process. I say we haven't done anything on the wash till now, and then we've looked at what happens for the phospholipids. So on the first injection, we can clearly see that we've got an abundance of phospholipids coming through, albeit with the higher molecular masses, they seem to have potentially been removed. It's interesting to note that as we then go on to the second injection, the difference in height for the 496 peak is minimal. There is an increase on the 524 peak, and the 524 peak seems to gradually increase as we increase the amount of plasma that we've injected going through that methodology. Similarly with the 704 peak, we see a small rise. It's fairly detectable. It's relatively low concentrations compared to the others. But what we do see a big difference is in the 758. And this may relate to some of the chromatography that we're observing. When we optimize the chromatography, we optimize it for the analytes. We've optimized it in this case for the 20-mer thymidine oligonucleotide, and we haven't optimized it for matrix components. Consequently, it's highly probable that some of these matrix components may not elute in that first injection. And I think that this is what's happening here. So we get a buildup of compound on the column, and they come off. Clearly, we need to do some further optimization on the wash steps, and that will come in a subsequent webinar. So I've alluded that there's more work. So what more work do we need to do? Clearly, we need to optimize the wash to try and optimize that matrix removal. For that, we can have a look at a range of different allotropic strength solvents in order to remove the matrix components. Reading the literature, it's very evident that adsorption is a big issue. We've already started performing some work in this area. Unfortunately, there just is not enough time to present all of the data, but at the next webinar, we will definitely be looking at doing this. We've also done some work on blowdown optimization and in particular reconstitution into the injection solvent and got some interesting data there, which again we'll present at the next one. The final stage that we really need to focus on in terms of developing the assay is looking at the optimization of the digestion step. So this is where we remove the oligonucleotide from any protein binding that could be occurring. And this involves a variety of different reagents, that all of which will need to be optimized. So I've given some of the reagents, and each of these will be involved in a particular step. So tris, TSEP, guanidine hydrochloride, and proteinase K. Finally, once we've got the final method, we'll then have a look at troubleshooting. And the idea behind all of this is to develop a general strategy. So not a unique method, but a general strategy which can then be applied to the analysis of oligonucleotides from a biological matrix. That concludes the presentation for today. What I'm hoping is that I've given you an oversight to how to start that analysis of oligonucleotides. Some of the challenges associated with it, in particular looking at the physicochemical properties and looking at log D, has really highlighted some of those. And then we've started to look at how to optimize that SPE process. There hasn't really been enough time to go through all of those optimization steps, but in the next webinar, we will be looking at how to optimize those remaining steps in terms of wash,
Hi, Tony, can you hear me?
Hi Gemma, how are you doing?
Hi. All right, sorry, I had some problems with my headset. Um, we do have time for a couple of questions.
Excellent.
The first, and thank you for a very interesting presentation. I like the stepwise approach to the development there.
Thank you.
Have you tried any other approaches for the SPE methodology?
Yeah, absolutely. It's a really good question for whoever sent that in. So we looked at using pH in order to control the state on the wax material. And this is the classical way in which you would use it for small molecules. But in this case, because the complexity, as I hinted at during the oligonucleotide description, it just really wasn't applicable. We actually had to use ion pairing reagents in order to drive the oligonucleotide off of the cartridge. But yes, we did have a look at different approaches. So I've kind of shortcut a lot of the method development. And even then, there was still a lot to go through. But yeah, good question. Thank you.
I think we've got time for maybe one or two more. So do you think that this approach would be applicable to a wider range of oligonucleotides? I mean, I know you said we were looking at maybe having a general method, but do you think other oligos would be suitable?
Yeah, the approach is very much I've been trying to encourage the team to do is not necessarily come up with one method does all, because I think with oligonucleotides, the complexity that we see within the physicochemical properties is such that actually it's an approach to method development that works most effectively, and that's what we've been trying to employ. So in essence, during the next webinar, we'll be looking at coming up with a flow chart that describes exactly how to go through that method development process. So that will become very optimal in the end. But yeah, it's an approach, not necessarily a specific method.
Brilliant. Just one more question. I know we're at the top of time. So why would you not use an SAX?
So a strong anion exchange uses a quaternary ammonium salt, and this forms a permanently charged surface, and the oligonucleotides seem to always have a charge on the surface, as we saw from the physicochemical data generated by chemicalized. So in that respect, it might not be applicable, and even the use of ion pairing reagents might not negate that charged interaction. So hence we went for a weak anion exchange. But a good question nevertheless. Thank you.
Brilliant. Okay, well, thank you very much again, Tony, for the really interesting presentation. If we haven't got to your question, please do keep sending them in. You can send them to webinar@avantosciences.com. There will be a recording made available within the next sort of 24 hours if you missed any of the presentation. And again, if you've got any questions, please do not hesitate to contact us, and hopefully see you all again at the next chromatography webinar. Thank you very much. Goodbye.
Thank you. Bye.